all these little arrows actually here indicate rotational flexibility. And the broken line here indicates that we also have a certain degree of structural flexibility within our Furanose ring system. So remember, Furanose is a 5-membered cyclic ring. Okay. And if we look at these individual bonds here, we see, okay, we have free rotational flexibility around the bond between the phosphorus and the oxygen here, and the phosphate, and then between the oxygen and this carbon here, and this between these carbons, and so on. And this is valid, okay, this assumption. Now, we also see that this arrow indicates free rotational flexibility around the N-glycosidic bond, which links the anomeric carbon to our nitrogenous base, whatever this base may be. And this is somewhat misleading because it was found that only a limited number of conformations can actually be assumed in DNA or are being assumed. And if we look at purine-based nucleosides and nucleotides, and these are exemplified here by adenosine, we see that there are actually only 2 conformations that are being assumed in nucleic acids and also in nucleotides, and these are called syn and anti conformations. And this is based on the position of, let's say, the majority of the purine ring system and the ribose with respect to the N-glycosidic bond here. So there's an angle here about which we can rotate this N-glycosidic bond, which is shown here as the Greek letter chi, okay? And in reality, we only have conformations where both of these moieties, the purine ring system and the deoxyribose, are on this, pretty much on the same side with respect to this bond, and we call it syn, pretty much together, okay? Or they are on opposite sides, as shown here for the anti-adenosine. Now, if we look at pyrimidine-based nucleosides and nucleotides, it's actually just the anti-conformation that is being assumed, and this is all based on steric reasons. Okay, so let's come to the conformations that can be assumed by the sugar, and these conformations have been called puckered conformations, puckered sugar conformations, and there are a variety of possibilities that this ring can, the structures that this ring system can assume. So this obviously is a representation of, in this case, deoxyribose, and we see the 5 atoms that make the Furanose ring system, the oxygen, and then the 1', 2', 3', 4' carbon, and the 5' carbon, which is outside of the plate, okay? And it's shown here as being above the plate. Here we have another representation. The difference between these 2 representations is that on the left side, the 2' carbon is out of the plane made by the other 4 atoms. The oxygen atom, 1' carbon, 3' carbon, and 4' carbon. And we see that in this representation, the 2' carbon is actually above this plane, so it's on the same side as the 5' carbon, and this conformation is called the endo conformation. Okay, so there are many terms in stereochemistry that pretty much describe a situation where 2 moieties are on the same side. We just saw syn. Now here it's called endo. Okay, don't ask me why. We'll see on the next slide where we have a table summarizing structural features of the different DNA conformations that we talked about, that this C2' endo conformation is actually what we find in the classical Watson-Crick double helix. So the classical Watson-Crick double helix of sugar conformation is such that this carbon number 2, the 2' carbon, is on the same side with respect to the plane made by the other 4 ring carbons as the carbon number 5. So this is our point of reference here. Okay. In theory, we could also have the 2' carbon being on the other side of the plane, but we don't necessarily find this in the conformations that we discussed so far. But if we just look at sugars, I guess this can also be assumed. On the right side of the slide, we see a conformation, pucker conformation, where the 3' carbon is out of the plane, It was shown in solid colors here is the situation where this carbon, the 3' carbon, is on the same side again as the 5' carbon. We call this the C3' endo conformation. Okay. So in principle, we could also assume the exo conformation where this 3' carbon would be on the opposite side. But again, taking a look at this table here, we see that we have the C3' pucker conformation actually being assumed in the A form of the DNA. So remember, the A form, just like the B form, the right-handed helix, and as I just indicated, I guess indicated again, can be assumed at low humidities. Okay, so let's take a look at this table, closer look, that summarizes the structural features here of the 3 types of conformations that we discussed for DNA, and we'll start here again, review what we talked about with regard to the classical Watson-Crick double helix. So remember, it's a right-handed helix. Our diameter is 2 nanometers, or 20 angstroms. It takes about 10, it says 10.5 base pairs actually to complete one turn in our right-handed helix. The helix rise is given here per base pair, so it's 0.34 nanometers or 3.4 angstroms. Remember, our pitch to complete one turn, if it takes 10 steps, would be 10 times that much, so 34 angstroms or 3.4 nanometers. The bases are not extremely tilted with respect to a virtual axis running through the molecule, so there's only a 6% tilt. Okay, so they're not completely perpendicular to a virtual axis, a little bit tilted. Just talked about the sugar pucker conformation, and we see that the glycosyl bond conformation is entire, okay, NT. So for both the purine-based nucleotides and, of course, the pyrimidine-based nucleotides, it's the anti-conformation that is being assumed to form this type of DNA conformation. If we look at the A form, there are slight differences. I talked about the somewhat wider diameter and things like this. The tilt is more extreme. Our sugars actually have changed to a C3' endo conformation, but our glycosyl bond conformations are still entire. Now it's quite different here if we look at the left-handed C form. So we talked about the narrower diameter here and things like this. The tilt is comparable to the B form of 7%. Then look at the sugar puckered conformations. It depends actually on what type of nucleotide you're looking at, whether it's a pyrimidine-based or purine-based nucleotide. So it's C2' endo for pyrimidines and C3' endo for purines. And also with regard to the glycosyl bond conformations, there's a difference whether it's a pyrimidine-based or purine-based nucleotide. So N type for pyrimidines and syn for purines. So quite a bit of local conformational changes have to occur to allow the molecule to assume a quite different conformation, because we changed it from right-handed to left-handed. Okay, that's what more extreme change could you actually do? So in general, I think most people simply assume that, okay, DNA in a cell, or maybe even in solution, would assume conformation that is somewhat comparable to the bDNA. And I guess for most situations this is rather valid assumption to make, but there are so many processes going on in the physiological conditions that require DNA to assume different conformations. And then also we usually assume that the conformations that are being assumed are somewhat independent of the nucleotide sequence, because we always see this pDNA molecule on pictures. But you may remember that I told you that the cDNA was first characterized for poly-GC DNA under experimental conditions. So obviously there is quite a bit of influence actually of the nucleotide sequence on structures, at least on local structures. And this is pretty much carried to an extreme here if we have self-complementarity within strands. Okay, and this type of self-complementarity comes in the form of so-called palindromic sequences. And you probably know what a palindrome is. What is a palindrome in English language?
It's like a word that's spelled the same going forward as backwards.
Okay, it's a word or a sentence that is spelled the same forward or in the reverse. Rotator, or nurses run, and they are more lengthy sentences actually than one could formulate. I always forget these things. My students come up with some occasionally, but so this would be a palindrome in the English language. Now here we are using this term to describe something somewhat different, obviously, namely sequences that are not overly long, a couple of nucleotides long usually, that are repeated in the complementary form within this strand. So if we look at this strand going from the 5' direction to the 3' direction, we see that we have a TGCGAT, okay, and then we have complementarity in the reverse direction here in this sequence, because this A is complementary to this T is complementary to this A, and so on. Now if we fold this molecule, these self-complementary regions, sequences can base pair and form a double-stranded stretch within this single-stranded molecule. Okay, so double-stranded is to be taken with a grain of salt here. We're still talking about one strand, but we have two regions of the same strand form double-stranded stretch, let's say. Okay. Now there is typically also a sequence between these two complementary sequences that then obviously can base pair with another complementary sequence that isn't there, okay? And these stretches then, these strands, parts of the sequences here, then form loops of varying lengths. Okay, so here we are talking about a relatively short loop, and this structure could be described as a stem loop structure, or in this particular case, because we're dealing with a relatively or very short loop here, we can refer to this as a hairpin. So very, very similar to the usage of the word hairpin in peptide structures or protein structures. Okay, tight turn pretty much. Obviously, if we are talking about DNA here, if we have self-complementarity in one strand, we also have self-complementarity in the opposite strand, okay, because the opposite strand would be complementary to the first one. And then we could form structures like this here that have been called cruciform. Now this obviously can occur in DNA, but it also can occur in RNA. If we have self-complementarity in RNA, we will form and easily form such stem loop structures, and we'll see examples of this when we talk about, for example, tRNA. So, let's come to another structural characteristic of DNA, and this is most easily explained if we look at a circular DNA molecule. And you may remember that I told you that all bacterial chromosomes are believed to be circular in nature, so they don't really have a beginning or an end, okay, just like a ring. And such structures can be described by a variety of parameters. For example, by counting how often these two strands in the double-stranded molecule wind about each other. Okay, and this is known as the linking number of L, which in this particular schematic here is 26. So this means that if you can actually count it, I did this, these two strands, these two individual strands in the double-stranded molecule are wound about each other 26 times. Okay, and this is not particularly interesting for us because we could also have an example where we have 55 or so. This 26 doesn't mean anything. Okay, but it's just an example here. And we assume that if we have a molecule here, like this one, that has a certain linking number, like 26, okay, it would be conformationally relaxed, okay? And in order to demonstrate what this means, I just ran up to get a very sophisticated model that I've been using for quite a while, doing a little bit of silicon tubing, okay, which I closed here, so just forget about this link here. But assume that this is a DNA molecule and assume that it's comprised of two individual strands. Okay, so if we are talking about a DNA molecule that has a particular linking number, L, and it is conformationally relaxed, it would mean that it would, for example, lie flat on the surface. Okay? Easy. However, if you actually increase or decrease the linking number, it means you overwind or underwind the DNA. Okay? Some people look quite bored, but this is actually of extreme importance for life. Okay? Then you induce conformational strain. Okay? It's as simple as that. Now I will try to simulate this. I cannot, since I only have one tubing here, but I can simulate actually what would happen if I induced conformational strain into this molecule. Okay, what would happen is that one way that the conformational strain could be accommodated would be if the double-stranded molecule actually winds about itself. Okay, and this process is called supercoiling. Okay, and if I induce more conformational strain, I could have varying degrees of supercoiling. Okay, so I could have supercoiling where these two strands are wound about each other actually a couple times, like this. Okay, and as you may have noticed also from my little model here, this also actually affects the volume of the molecule, which has ramifications for molecular biological work. So I'll point out in a moment. So here are micrographs of a bacterial chromosome, I think from E. coli or so, which show different degrees of supercoiling of such chromosomes. So in the first slide, obviously, we are dealing with something that is probably quite conformationally relaxed, and then we have varying degrees of supercoiling. Now, who does molecular biology here in this class? Anyone working for Dr. Horn? You do that? Do you work with plasmids? Not yet? A little bit. Okay, so what you do often in molecular biology is that you transfect cells, which means that you actually incorporate foreign DNA or something that you usually want to express. Okay, so if, for example, if we in my group wanted to express certain antibody binding sites in E. coli, we would actually utilize plasmids. These are relatively short circular DNA molecules. We find bacteria that actually have information often for things like antibiotics and so, and these have been used as so-called vectors in molecular biology to infect pretty much other cells and to introduce this foreign genetic information to the cells, and then the protein biosynthesis machinery and transcription machinery of these organisms would be utilized to express proteins whose information is stored on these vectors. Okay, so there are different vectors that you could use, viruses or these plasmids. And what you do is that you take a vector and then you cleave it using restriction endonucleases. Okay, at certain sites, which often enough are palindromic in nature. Okay, and then you insert a gene of interest, and then you ligate the DNA again. And I don't have another piece of tubing here, but actually your new plasmid would be longer. Okay, and since you increased the molecular weight of this molecule and the length of the molecule, you could actually test whether or not you have successfully introduced your gene of interest into the plasmid by doing a gel electrophoresis experiment. Okay. Now, gel electrophoresis is based on the fact that you have a certain relationship between the volume charges you have in a molecule and the electric mobility of this molecule. Okay. So in molecular biology, you assume that certain lengths of nucleic acid molecules are related to the electrophoretic mobility in an electric field. Okay? But this is based largely on the size of the molecule because we have more or less even distribution of charges in these molecules. Okay? So, if you use plasmid circular DNA that can actually assume a variety of different conformations based on supercoiling, you may not be able to see on a gel then that you do have the electrophoresis whether or not you have successfully inserted your gene. Why? Because your molecules can assume a great variety of different volumes which are not necessarily representative of the length of the molecule. Okay, so there is no clear relationship between the length of the molecule and the volume it assumes and the time it takes to move through an agarose gel. And instead of getting in gel nice bands like this, you may get all kinds of seams. Okay. So, how could you actually address this problem? What do you think? Usually the students who have done this, but just cut it. Okay, so you apply a restriction enzyme to cut your molecules in a certain position, and then they will not be circular anymore, and you can analyze them, and then you should actually get reliable results. So if you ever come to a situation like this, where you do this type of molecular biology experiments and you get things like this, remember this class. Okay? So, and we have quite a bit of experience in that. Often enough, as students come to us, don't know what's going on. In most cases, it was exactly that. Okay, so we just talked about the accommodation of conformational strain by supercoiling. Another way to accommodate conformational stress, which I cannot show with my little model here, is the local strand separation. This is of greatest importance here for our purposes and for life, because what happens here is that when DNA molecules are overwound or underwound, and we could have this kind of supercoiling, we can also have a local strand separation as shown at the bottom of the slide. So here our linking number, I guess, would be 8 turns, where we would have a relaxed molecule. If we underwind it, okay, which is quite common actually, that DNA is underwound, we induce conformational strain. We could do supercoiling, or we could have a local strand separation. So why is this local strand separation of such importance? Because in order for proteins such as DNA or RNA polymerases to access individual strands, we have to separate the 2 strands that are hydrogen bonded in double-stranded molecule. Okay. So we will see when we talk about transcription and translation in a little bit more detail that in the so-called initiation phase, which describes how the polymerization machinery comes together and starts doing the polymerization process, that we always have a separation of a double-stranded DNA molecule, whether it's replication of DNA or whether it's transcription. Okay. So there are enzymes that can actually induce conformational strain, and there are enzymes that can remove conformational strain. So of importance in particular are here so-called DNA topoisomerases. Put away my little model. So this is a let's say, a little bit outdated representation of how a topoisomerase of type 2, which can also be called DNA gyrase, works. And it's a very, very oversimplified point of view, but still like to show this because it pretty much shows what I just did here, how conformational strain can be induced by just wrapping DNA molecule about something. Oh, that's not very good here what I did. No, okay, the model doesn't work anymore. But I did it before, we saw that out. Okay, like this. And then could also be removed by simply cutting it. So topoisomerase type 2 cleave two strands in the double-stranded molecule, and then, as we see from this picture, actually use or remove strain here by leading or guiding the two ends of this cut part of the molecule to the other side of whatever was wrapped about here, about this topoisomerase. Okay, so we have these coils here, then we cut, and then we lead this part to the other side and ligate again, religate. Now nowadays it's known that this is a little bit more complex, this process, and this is a picture, I think, from the newer editions of the textbook that we are using. So we have different so-called gates to which parts of the molecule, double-stranded molecule, bind. But pretty much the same thing happens at one point, namely that we cut one part of the double-stranded molecule and then lead it through the other side and then religate it. And we see that this is an energy-requiring process. What is interesting here actually is to take a look at what happens in the active side of the topoisomerase, because we have tyrosine residue that is of crucial importance here. Because this tyrosine residue, actually let's just focus on this here. This tyrosine residue, oxygen of the phenol here, attacks the phosphodiester bond and forms a transient covalent bond here with one portion of the sugar phosphate backbone. So we have successfully cleaved here the DNA molecule, and then we have a religation when this end attacks this phosphorus again, and we ligate the strand again. Okay, so tyrosine residue. What we're going to do now is take a closer look at particular eukaryotic chromosomes for the time being, and take a look at the packaging. But just as a reminder, we see here E. coli and stained with a fluorophore here the DNA molecules, the chromosome that we have in these cells. So each bacterial cell, like in E. coli, just has one chromosome. We talked about this. We saw that the length in base pairs is somewhere in the range between 4 and 5 million or so, if I remember correctly. But this is one circular DNA molecule. Okay, it's believed that this circular DNA molecule is associated with proteins and somewhat attached to the inner side of the plasma membrane, the cell membrane. But remember that prokaryotes do not have organelles, so this is just in the interior of the cell, it's not in the nucleus. The situation is quite a bit different if we look at eukaryotic cells, okay? And now the eukaryotic cells have a nucleus, and at least when the cell is not dividing, the majority of the DNA that we find in a cell will be found in the nucleus. Okay, so we also find DNA in mitochondria, and if we're talking about plant cells, also in chloroplasts, plastids. But if we just look at animal cells, we will have DNA in mitochondria, but the majority of the DNA of a cell will be in the nucleus. And if you look at a cell here in the nucleus, on the microscope, you'll see different areas that have different densities. So here we have a large blob, which is rather dark, rather dense, and that's the nucleolus. And
And that's the area where we have assembly of ribosomes, okay? Transcription of RNA and assembly of ribosomes. And then the rest pretty much of what we see here, which we can refer to as chromatin in general, has lighter areas and darker areas. And the lighter areas have been called euchromatin, U for true, okay? And these are believed to be areas where we have transcription going on, okay? So they're genetically expressed, while heterochromatin are the denser areas, and okay, we'll see for reasons we're going to discuss that we probably do not have a lot of transcription things like this going on. What I want to point out is the scale here. So you can just guess how big this cell is, and just to put this in perspective, the typical diameter of an erythrocyte, so red blood cell, is somewhere in the range of 7 or 8 microns. Okay, and they're, depending on the cell type, there are great variations with regard to the lengths of cells. Muscle cells, for example, can be very long. Some nerve cells are extremely long. Okay, but average size of a cell, like an erythrocyte, 7 to 8 microns in diameter. However, this may pose a problem, okay, because human chromosomes, okay, if we look at just humans here, are between 48 and 240 million base pairs. Okay, so if you calculate the lengths of these molecules, if they were fully extended, you could actually calculate that we have a size variation of these fully extended molecules, okay, somewhere between 1.6 and 8.2 centimeters. So we're talking not that much, okay, 2-3 inches. I can see that, right? Okay, I can't see 8 or 10 microns here. But hey, so how do we get such a long molecule into such a tiny cell, in such a tiny nucleus? Okay, by condensing the molecule. And that's a process that is not fully understood, but consists of various levels, which are summarized here, which show that starting with a fully extended DNA molecule, which is hypothetical here under physiological conditions, okay, because it wouldn't fit into a cell, we first wrap it about a structure that is made of proteins, okay, and we actually form something that is called nucleosomes, which have a diameter of about 11 nanometers. And then we wrap these structures in a certain way, like we were braiding these things to produce something